Most receptors we come across in pharmacology are protein-based. Why is this so? Let's explore this in Learn Efficient Pharmacology. Pharmacodynamics Part 4, Receptor Conformations. Remember the structure of proteins from our biochemistry lessons? Amino acids are the building blocks of proteins. In an amino acid, there is an amino group and an acid or a carboxyl group. The amino group of one amino acid combines with the carboxyl group of another amino acid to form a covalent bond. This is called a peptide bond, and the resulting linear sequence forms the primary structure of the protein. These peptide bonds have a partial double bond character. This is pretty rigid and will not allow for much rotation. But the alpha carbon atom of the amino acids forms single covalent bonds with nitrogen of the amino group, carbon of the carboxyl group, and side chains. Unlike the partial double bond, these single bonds allow for rotation. When rotation occurs around these single covalent bonds, the primary structure adopts a three-dimensional shape called the secondary structure. One common shape thus formed is when the peptide chain winds around like a spiral staircase or helix. Since this was the first secondary structure to be discovered, it was called the alpha helix. The alpha helix is stabilized by hydrogen bonds formed between the oxygen of the carboxyl group of one amino acid and the hydrogen of the amino group of another amino acid, that is 4 residues ahead in the sequence. The peptide chain folding does not end here, and secondary structures can be further arranged in space. For example, 7 alpha helices can be arranged in a tightly packed bundle forming a 3-dimensional structure. This is called the tertiary structure of the protein. This tertiary structure should also be stabilized. This is done primarily by non-covalent forces. You might remember that in our very first video on passage of drugs across the plasma membrane, we had seen how drug molecules diffuse without using any additional energy, but just the kinetic energy of the solvent molecules derived from heat in the system. Similarly, owing to transfer of thermal energy within the system, some of these weaker covalent linkages can break. This changes the 3-dimensional structure. This changed structure is stabilized by new interactions. So the same protein can exist like this, or like this, or like this. In fact, a protein can be arranged in numerous ways in space. Each arrangement is called a conformation. Thus, proteins are flexible and can shift from one conformation to the other. One might be an inactive conformation, whereas another might be an active conformation that triggers downstream pathways resulting in cellular effects. This flexibility in conformations is one of the most important reasons why proteins are suited as receptors. So, if a huge number of conformations are possible, does a receptor go through all of them? We will see that in the next video. Here is a summary of what we discussed. Here are some good to know facts. This is a must-know fact.